Can you hear me?
Yes, ma'am.
Yes. Yes.
Okay. All right. So thank you. Okay. Assalamualaikum. Basically, okay. So for our lab session today, I hope it should not take too long. Although we are going to cover for two experiment. Okay. So for today's experiment, we are going to look at two experiment, which is experiment 2 and also experiment 3. Okay, so for experiment 2, we are going to look at video demonstration, lab demonstration of cell staining. Okay, so in the title, it says here, still cell staining and also measuring cell size. So basically, we are going to look only on the part for cell staining. We are not going to look at measuring cell size. Okay, so only cell staining. Okay, so the objective of this experiment, the for the second experiment, okay, so is to prepare a wet mount of plant cell and animal cell. Okay, so the plant cell that we are going to be observing is from onion. Okay, the epidermis of onion. Okay, so plant cell, okay, plant cell, as you know, they are surrounded by cell wall, and then because due to the presence of cell wall, you will observe later that plant cell will have a fixed, right, a fixed rectangular shape. Okay, and then within that plant cell also there is the presence of a large central vacuole, and then when you are observing the plant cell under the microscope, you will notice that the nucleus the nucleus of the cells will be located at the peripheral side of the cell, okay, due to the presence of this large central vacuole. So it means that the other organelle also will be pushed to the side. Okay, so but because we are using light microscope, okay, uh, only large organelles, only large structures of the cell can be observed. Okay, so it means that under the light microscope, you should be able to observe the structure of the plant cell wall, the presence, okay, the presence of plant cell wall, the plasma membrane, um, the effects of having this large central vacuole. Okay, you can see the effects of this, the presence of large central vacuole, and also the presence of a nucleus, okay, in every cell. Other organelles are not present, for example, chloroplast. Okay, so because we are observing uh, um plant cell, okay, from the epidermis of onion. Okay, the epidermis of onion. So onion, as you know, they are found in the soil. Okay, in the soil, and so they are not exposed to the sunlight, and so this for, and therefore the onion, the bulb part of the onion will not be carrying out photosynthesis. Okay, will not be carrying out photosynthesis. So that's why when we observe the plant cell using this onion epidermis, you will not be able to observe the presence of chloroplast, although it is a plant cell. Okay, it is plant cells. So there's no chloroplast as the specimen is from the bulb of onion. Okay, but if we were to observe plant cell from leaves, okay, plant leaves, the leaf part of the plant, so you should be able to observe the presence of chloroplast. Okay, so under the microscope, okay, under light microscope, you should be able to observe chloroplast also. But if we were to use, the leaf part of the plant. Okay, so other organelles such as the Golgi apparatus, the mitochondria, the ribosomes, the all that organelles you cannot see because due to their small size, and the light microscope cannot um, cannot let you see all these smaller organelles. Okay, So and then okay, so and then as you can see here, okay, one of the materials that we are going to use are dye, okay, dye stain. So the stain that will be used is iodine stain and also methylene blue. Okay, so the purpose of using this stain is so that the stain will stain these cells. Okay, the cells of the allium species, the epidermis, and also the chick cell. Okay, so if you do not, okay, dye the plant cell using this stain, you cannot observe the cells clearly. Especially you cannot observe the presence of the nucleus. Okay, so that's why you need to dye this cell using dye, okay, iodine stain and also methylene blue, so that you can observe clearly the presence of the nucleus, especially the nucleus. All right. Okay, if you look at the first objective, to prepare a wet mount. So why do we call it as wet mount? Because we are using the stain. Okay, so you have the microscope slide. Okay, the microscope slide where you place your specimen, the plant cell, and also the chick cell, and then on top of that cells, the specimen, you are going to drop a few drops of the dye. Okay, so and then you are going to cover with cover slip before you place your specimen, your slide that has your specimen onto the microscope stage. Okay, so that's why we call it as a wet mount. So any excess liquid, okay, that are on the microscope slide, you have to wipe off. Okay, so the purpose of wiping off this excess fluid, so that when you are observing under the microscope, this excess fluid from the iodine and also the methylene blue will not touch the lens and causes the lens of the microscope to be contaminated. Okay, so remember that. So walaupun you all tak buat eksperimen ni, itu adalah asas kepada penggunaan mikroskop ni. Okay, so make sure when you are observing your specimen under the microscope, your microscope is clean and dry. Okay, so that any excess liquid, okay, on the specimen will not touch the lens of the microscope. So bila awak buat, if you were to do it practically, kan, nanti kalau masuk lab ke whatever, so make sure you do that. Alright. Okay, so the apparatus. Okay, so the second objective, to compare stained plant and animal cell under the microscope. So after you have placed your specimen onto the microscope stage, you are going to find the right focus using the coarse and fine focus until you get a sharp image of the specimen, and then you are going to compare the difference in the structure of the plant and also animal cell. Okay, so for animal cell, because they don't have any cell wall, so basically the shape of the animal cell will be spherical. Okay, and the nucleus of the animal cell will be located roughly at the center of the cell. Okay, so later we are going to look at the demonstration video. So saya briefing dulu, right? I'm going to brief to you first, okay, to get you to be able to picturize, okay, the experiment. Okay, so and then the third objective is to observe, prepare prokaryotic cell using immersion lens. Okay, so you have done your video of you using the virtual microscope. Okay, and then you have watched the video of how to use the microscope by Miss Sabrina. So you should be able to know by now that for the light microscope, it has 4 lenses. Okay, you have the 4 times lens, 10 time lens, 40 times, and also 100 times lens. Okay, so for the 100 times lens, okay, you cannot just observe it like that. You have to drop a drop of immersion oil onto your specimen, and then only you can observe using the 100 time magnification. All right. So usually in the lab, for the prokaryotic cells, okay, the source of getting this prokaryote, usually in the lab, we use yogurt, okay. Okay, so the apparatus, scalpel, toothpick, forceps, microscope slide, microscope, cover slip, graticule, and also prepared slide. Because we are not doing this part of the experiment, measuring cell size, so we will not be needing any graticule. So graticule is basically um, um, an apparatus used to measure the size of specimen under a microscope. All right. So I'm not going to go through the procedures. We are going to watch, okay, the demonstration video of this experiment prepared by Madam Liza. Okay. So I'm going to share with you the video.
Welcome to Acid.
Kalau tak boleh dengar video ataupun tak boleh nampak, you all bagi tahu. So I'm going to play the video right now.
Hi everyone, welcome to Acid Biology YouTube channel. Today we are going to watch a demo video on Experiment 2, Cell Staining and Observation. In this experiment, we will observe 3 different types of cells, which include plant cells, animal cells, and bacteria. We will first run through the materials and apparatus that we are going to need. Next, we proceed with preparation of slides and staining process. Finally, we are going to observe the slides under the microscope using the proper technique. But before we continue, don't forget to like this video and subscribe to this channel. Thank you. So, let's continue with our experiment. So first of all, let's start with the materials and apparatus needed. There are 3 materials needed in total. They are onion, iodine, methylene blue. 6 apparatus to complete this experiment. The first one is scalpel, forceps, cover slip, microscope slides, toothpick, and a microscope. Now, let's start preparing the slides. For plant cells, we are going to use onion epidermis and iodine as staining.
Risau si manja tak dapat cukup khasiat, berikan Sustagen. Sustagen, 100% khasiat dan lazat.
Cut a small section of an onion using a scalpel. Use forceps to remove the cutting. And carefully peel off the thin, transparent epidermis. Transfer the epidermis in the center of microscope slide. And then put a drop of iodine stain on the epidermis. This is a wet mount. Place one edge of cover slip to one side of stain and slowly lower the cover slip. If you need to remove excess stain, touch the edge of slip with paper towel. For animal cells, we are going to use chick cells and stain them with methylene blue. Use a toothpick to gently scrape inside of your chick. Then smear and mix the cell in a distilled water. Next, put a drop of methylene blue to stain the cell. Place one edge of cover slip to one side of the stain and slowly lower the cover slip. Now, let's continue with slide observation. You will learn the right way to use a microscope. So, make sure to set 100% attention to the demonstration. We will use the plant cell as example. Microscopes usually are stored in a dry cabinet. If you need to get the microscope, be sure to hold it right. Unwind the electrical cable and turn on the electrical power of the microscope. Unscrew the ocular lens and remember to adjust it to the placement of your eyes. Switch on the microscope and remember, do not use the fine focus knob. Use coarse knob to adjust the stage so that the stage is at the highest position. Place the specimen slide on the stage and secure with slide holder. We will start off with alien slide. You can see the stage knob can be adjusted to move the slide front, back, or left and right. Look through the eyepiece and make sure not to use fine focus knob at this moment. Move the coarse knob until the image comes into focus. And then you can use the fine focus. Make sure that the image is clearer. This is what you will see under the microscope. Change to the next objective lens. Once 10 times objective lens is clicked, adjust the sharpness and clarity using only fine focus knob. Please remember that at this point, you cannot use coarse knob. You need to adjust the condenser and light intensity until the image is clear and bright. This is what you will see under the microscope at 100 times magnification. You can use the stage knob to move the microscope stage so that the specimen is in the center of the field of view. Do the same with the 40 times magnification power of your objective lens. Please remember to only adjust fine focus knob. Remember, you cannot adjust coarse knob at this point. Again, you can adjust the light intensity so that the image is clearer and brighter. This is what you will see under the microscope at 400 times magnification. You can adjust the stage knob so that the image is at the center of your field view. If 1000 times magnification is required, this is what you need to do. After getting a clear image at 400 times magnification, turn the barrel of the microscope so that the 40 times and 100 times lens are halfway between and directly over the slide or cover slip. Next, you need to place a drop of oil in the middle of the slide or on the cover slip. Carefully turn the objective lens until the 100 times magnification lens contacts the oil and snaps into position. You can adjust sharpness and clarity using fine focus knob. After the observation, remember to wipe off the immersion oil and clean the objective lens with lens tissue. We already observed our plant cells under the microscope before. Now, let's see how animal cells look like under the microscope. This is chick cell under the microscope at 40 times magnification. This is chick cell under the microscope at 100 times magnification. This is a chick cell under the microscope at 400 times magnification. This is how bacteria looks like under the microscope at 400 times magnification. The bacteria is circular shape and it is stained in purple. Well, students, that's all for experiment 2. Do complete your observation and don't forget to submit your lab report on time.
Bekalkan di rumah. Bekalkan tenaga. Bekalkan di rumah.
Bekalkan kebaikan Milo di rumah.
Hi.
Okay, so that's basically the demonstration video for experiment 2. So okay, so for your lab report, right? So like the lab report that you have done for experiment 1, so you must have the title of that experiment. So the title, you just omit out the measuring part, and then I don't make it compulsory for you to do the abstract for the lab report, but for the report of your mini project you must include abstract. Okay, so yang untuk report, kalau awak nak enhance your ability, your your ability to do an abstract, so then you can do an abstract. Okay, so the abstract is basically the last part of the report that you do after you have your result, discussion, conclusion. Then from there you can write the abstract, okay, which is a summary for the whole report. All right, so abstract you can do or not do, it's up to you for the report. All right, for the lab report of the experiment, tapi untuk mini project you have to do the abstract. Okay, so next you have to have your introduction. Okay, so your introduction should not be too long. Okay, about 1 page, 1 or 2 page. Okay, 1 page. Okay, so 1 page. Okay, and then after that, after the introduction, you rewrite the objective, okay, of the experiment, the three objective that that we looked at earlier, and then you list down, and then the next part is the materials and method, the methodology. Okay, so you list down all the materials and the apparatus needed. So just leave out the graticule part, and then after that is the procedure. Okay, so the procedure must be written in past tense, okay, because it is a report. Okay, you are reporting the work that have been done. Okay, so you have to write in past tense, and then the result part. Okay, so for the result part for this experiment, I want you to draw the observation. Okay, you have to draw the cell observed under 3 magnification. Okay, so which is under 4 time magnification, 10 times magnification, and also 40 times magnification. Okay, so you have to draw for each animal and plant cell 3 drawings under 3 different magnification. So if you notice, okay, you know that the microscope has 4 lenses, 4 times, 10 times, 40, and 100. When you are observing a specimen under a microscope, you always use the lowest magnification first. Okay, so that when you are observing your specimen under the lowest magnification, you have the whole field, okay, of the specimen to observe. Okay, the whole field. So you know that where to focus. Okay, when you increase the magnification to 10 and 40. If you straight away use the highest magnification, then you will have trouble finding the right place, okay, to observe, finding the right the clearer, okay, the much more nicer region to observe if you just go straight using the 40 times magnification. Macam awak guna Google Map lah kan, awak nak cari satu tempat, awak terus pergi zoom ke kan. So awak akan tengok dulu the whole map, then baru you find the place that you want kan. Same goes to the microscope. Okay, so you use the lowest magnification first so that you know where to observe when you increase the magnification, right? So and then okay, so if you notice that Madam Liza, when she showed you the image, okay, the specimen image for each magnification, she says, okay, the term 40 times, 100 times, 400 times and 1000 times. Okay, so mana datang yang banyak-banyak magnification ni, sebab awak dah kalikan dia. Kalau 4 times lens, dia akan jadi 40 times. Okay, the 10 times lens, dia akan jadi 100 times magnification. The 40 times lens will become 400 magnification, and for the 1000 time lens, dia akan jadi 1000 magnification, right? Okay, so for your drawing, okay, I want you to draw the specimen, not just take a screenshot and then you print out and then you include in your report. Okay, I want you to draw the image, okay, of the specimen observed under this different magnification. Okay, so you must have the ability to draw the specimen. So you observe and draw, observe and then you draw. Okay. Sekejap. Alright. Okay. So this one is not the clearer. Okay. Okay. So so this one you should you can use this image to draw your observation under 40 times magnification. Yeah, 40 times magnification using the 4 times lens. Okay. So as you can see here, okay, in this image, right? You can observe that the plant cell is rectangular in shape because due to the presence of the cell wall. Okay, so these are the black line here are the cell wall, and the black region here, okay, it is actually not part of the plant cell. It is actually air bubble formed, okay, when you are preparing the slide. Okay, so if you notice that after you have placed the specimen under the microscope under the microscope slide, and then you place your specimen and then you drop the dye on top of the specimen and then you cover with cover slip. The process of covering the specimen with cover slip will lead you to form air bubble. So that's why bila step yang dekat sini ni, okay, you have to be very careful, okay, so that there's less formation of air bubble. So as you can see, Madam Liza will place the cover slip on top of the specimen, and you have fluid, okay, underneath. So the presence of the fluid under on the specimen will lead to the formation of air bubble if you place the cover slip too fast, okay, too fast. So it will lead the formation of air bubble even more if you just drop it like that. Okay, so the ideal way is that you should get for example a pen or a knife to support the cover slip, and you let it drop really slowly to make sure that there's less formation of air bubble. Okay, so okay,
Do the same.
okay, so this is under 40 magnification using 4 times lens. Okay, so yang hitam ni it is not part of the structure of the plant cell. So you just for your drawing, you take the much more nicer region for you to draw. Okay, so and as you can see, here is the nucleus. So if you if you don't stain your sample using the iodine, you will not be able to observe the presence of nucleus. And as you can see, the most of this nucleus are at the edge, okay, at the edge of the cell. Okay, so this is due to the presence of large central vacuole within the cell. Okay, so make sure you label all the structures. So you have to label the cell wall, and underneath you have the plasma membrane, and then inside you have the cytoplasm, the nucleus, and then the presence of large central vacuole. Okay. So for the 10 times magnification also you have to draw. Okay, so this is under 100 times, okay, 100 times magnification using the 10 times lens. So ada yang lebih clear tak? Okay, any good, right? So as you can see, so this is observed under 100 times, like 100 magnification using 10 times lens. So this is the nucleus, alright, the cell wall, and then yang warna kuning sikit ni, cerah sikit ni is the plasma membrane of the cell. Okay, is the plasma membrane of the cell. So inside you have the cytoplasm, and then the presence of the central vacuole, you cannot really see structure tu sejelas macam nucleus. Okay, sebab nucleus tu dia ada komponen, dia ada kromosom kan. So the dye will stain the nucleus, right? But for the central vacuole, the content mostly is water kan. So that's why you cannot see really clearly the central vacuole, but you can see the effects as you can see. Here, the nucleus is at the peripheral of the cell. Okay, so this may be the region of the central vacuole, and these are the air bubble formed. Okay, so for the 400 times magnification, ini 400 ke? Sekejap ya. Okay, tadi 10. Okay, 10 times lens. So next is the 40 times lens, which is 400 magnification. Okay, so this is the clearer image. And tadi tu, yang ni, these are the air bubble. You take the image with the much clearer view. Okay, so this one, as you can see, you omit out the air bubble. Okay, so you can see the nucleus, the plasma membrane, the cytoplasm, and here is the region of the central vacuole. Okay, so nampak the presence of the central vacuole will push the nucleus to the side of the cell. Alright, so yang hitam ni adalah cell wall. Bawah tu you have the plasma membrane. Alright, for the okay. Okay, so for the chick cell, so this is the image observed under 10 time lens or 100 magnification. 40. Okay, 40 magnification using 4 times lens. Okay, 40 magnification using 4 times lens. Yang paling rendah. So you see that the cell is smaller. So this is a chick cell. Okay, so chick cell, human cell, they are circular in shape and they are squamous because we are taking sample from the chick cell. Kita dah belajar kan animal tissue. So animal tissue, we have 3 different types. Okay, you have squamous, you have cuboidal, and you have columnar. Okay, and then because we are taking sample from the chick cell, it is obviously squamous. Okay, squamous epithelial cell. Okay, so so as you can see, that's why the cell is circular and flat. Okay, flattened, and then the arrangement of the cell lining your oral cavity, the chick cell inside, is arranged in layers. Okay, the arrangement of cell in layers, we call it as stratified. Okay, stratified squamous epithelial cell. Okay, it forms the tissue of your oral cavity, right? Okay, so this is observed under 40 magnification using 4 times lens. Okay, yang paling rendah. Okay, so this is observed using 10 time lens, okay, 10 time lens, which is 100 magnification. So you can see that the cell is much more clearer, bigger, and nicer to observe. So you have several of the chick cell here, and you can see that at the center of the cell you have the nucleus. Okay, so that's why we are using the stain to stain the nucleus, right? So you just label the structures of the cell. So you have the plasma membrane, inside you have the cytoplasm, and then you have the nucleus, right? Okay, so this is the image observed under 40 times lens, which is 400 magnification. So you just draw this one cell and then you label. Okay, you label every structure. So remember, although you are drawing the same structure, plant cell, animal cell, every time you draw your drawing, you must label. Okay, you must label the structure. So the plants, so the animal cell, you have the plasma membrane outside, okay, as the barrier for the cell, and then you have the nucleus at the center, and inside you have the cytoplasm. Okay. So this is the prokaryotic cell observed under 1000 magnification using 100 times lens. Okay, so as you can see, the bacteria is circular in shape or spherical. Okay, or bacteria that are spherical, we call it as cocci lah, group dia cocci. So bacteria ni bergantung pada shape dia. So ada yang spherical, ada yang spiral, ada yang rod shape kan. So the shape of the bacteria will lead to the name of that bacteria nanti kan. So itu nama-nama bacteria tu we are not focusing at this point, alright? Okay. So that's for your result. Okay, so when you are drawing your observation, I want for every drawing, okay, it is not less, it must be half a page big. Okay, half a page at least half a page big. Saya tak nak draw dia comel je kat situ kan, kecil je. Half a page, okay, for each drawing. Okay, at least half a page. Okay, so you must draw your observation on a plain piece of paper. Okay, plain piece of paper. So I don't mind whether you want to draw using pencil on paper, or you can use your gadget lah kan. Kalau ada siapa yang ada tab ke iPad ke, you can draw using those gadget. Asalkan you draw your observation. Okay, not taking someone else's picture, not taking the image from the internet. So you must draw. Okay, I trust you that you must draw your image using your own effort, right? Bagi-bagilah, you have 5 members in a group, so you bagi-bagikan, so then you combine, check before you submit, right? So because the work of your member will influence your mark as well, right? Okay, so for the discussion part, I want you to discuss your result based on theory lah kan. Discuss your result based on theory. You ceritalah kan the structure of the membrane, the structure of the cell and all that. Alright, so discuss result based on theory. So your discussion should be like 1 or 2 page. Okay. And then you conclude your observation. Yeah, for conclusion, you answer back the initial objective of the experiment. So every time you are writing a conclusion, okay, you must refer back to the original objective of the experiment. So you answer back whether you managed to achieve or not the objective of the experiment by summarizing the result. Okay, the result and the discussion in the conclusion part. And then for the reference, I want you to include roughly about 2 or 3 references, okay, that you write according to the proper way of writing reference. Okay, so so far, do you have any question on experiment 2 before we proceed to experiment 3? Okay, so no question. I hope you know what to do for your report. Okay, so I'm going to share the next video. So macam iklan pagi ni ada iklan Milo lah, aiskrim McD lah. So saya tahu ada yang tak sarapan lagi, saya pun tak sarapan. So after this you can take your breakfast. Okay, so I'm going to share with you for the experiment 3. Okay. So kita apalah, kita tengok dulu video.
Experiment 3. Investigating cell membrane permeability using turmeric, Curcuma longa. This is the last experiment for this semester. In this experiment, we are going to investigate the effect of ethanol on the surface of cell membrane of Curcuma longa. Now, let's take a look at the materials needed in this experiment. We need turmeric or Curcuma longa, ethanol, and distilled water. Now, let's take a look at the apparatus needed. We need cork borer, scalpel, ruler, forceps, beaker, and measuring cylinder, not to forget a white tile. Use a cork borer to cut 6 cylinders of fresh Curcuma longa tissue. Hold the turmeric with your left hand tightly, and by applying force, I mean a lot of it, start the process by pushing down the cork borer through the tube. Take out the borer and then use the solid pin to remove the fresh cylinder tissue out of the borer. Use a ruler and a scalpel carefully to cut the cylinders so that they are at the same length. You can clearly see in this video we use 2 cm measurement for each cylinder. Cut it gently and place all cylinders in a beaker of water. Let the cylinder soak in water for 5 minutes. Make a 25% dilution of ethanol. Total volume needed is 10 ml. So, in this video, we use 7.5 ml of distilled water and 2.5 ml of ethanol. Label 3 small beakers respectively. One with 100% ethanol, second one with 25% ethanol. As you can see, we are mixing the dilution now. And finally, a beaker labeled distilled water. Fill each beaker with its respective solution of 10 ml each. Remove all cylinders from water and place onto a paper towel to dry. And then place 2 cylinders in each beaker. Let stand for 15 minutes. After that, we observe the color change of the solution in each beaker. This is the end of this experiment. Now, you need to record all your observation in a datasheet experiment. A datasheet must consist of a table to record color change of 3 different solutions used. Remember, this is the initial color of 100% ethanol, 25% ethanol, and distilled water. They are all colorless. After 15 minutes, the solution changes in color. This is 100% ethanol, this is 25% ethanol, and this is distilled water. Describe the color. You may also put this figure in your datasheet experiment. Finally, you need to discuss all of the findings. Why do you think the color changed? What happened to plasma membrane in reaction to alcohol? After completing your lab report, don't forget to submit it in your Google Classroom on time. Good luck!
Okay, so that's the video for lab demonstration of experiment 3, investigating cell membrane permeability using turmeric. Okay, so basically you know that the color of turmeric is yellow. Okay, so the color of the turmeric is yellow due to the presence of pigment within the cell of the turmeric. Okay, so when you are writing your discussion later, I want you to find out, okay, what is the name of the pigment that caused the uh, the the turmeric to be yellow in color. Okay, the name of that pigment. Okay, so you do your reading, okay, to find out the name of that pigment and where does this pigment is stored in the cell of the turmeric. Okay, so usually, okay, all these pigment are stored in an organelle. Okay, so organelle within cell are surrounded by membrane. Plant cell also are surrounded by plasma membrane. Okay, so if you look at the title of this experiment, investigating cell membrane permeability using turmeric. Okay, so in the procedure of the experiment, you have to prepare cylinders of turmeric, and then firstly you have to immerse the turmeric in distilled water. Okay, so bila awak tengok kat dia punya procedure ni, saya just rewind back. Okay, fast forward. Okay, so you are preparing the turmeric cylinder using the cork borer. Okay, and then okay, on this stage you are going to immerse first, okay, the turmeric in distilled water. Okay, so the purpose of immersing this turmeric in the distilled water first is so that when the first procedure where you prepare the turmeric cylinder using the cork borer, you are going to damage, okay, the cell of the turmeric on the outside here. Okay, so the damaged cell will release the pigment from the cell. So that's why you need to immerse first the turmeric inside distilled water so that the pigment on this damaged cell will be released first. Okay, the pigment on the damaged cell outside here, okay, will be released into the distilled water. So you don't want any damaged cell to be placed in the alcohol later. Okay, so you want to make sure before you place the cylinder inside alcohol, all the pigment from the damaged cell is released into the water first. Okay, so you immerse the cylinder in distilled water for 5 minutes if I'm not mistaken, and then you take out okay, so this one you preparing the alcohol. Okay, the distilled water, the 25% ethanol, and also the 100% ethanol for you to immerse the cylinder later. Okay. Okay, so this stage, after you have take out the cylinder from the water, the initial water that you immerse the cylinder, you placed it on the tissue paper to wipe off excess water and also the excess pigment that are left. Okay, on the outside part of the cylinder. Okay, so nampak Madam, this is Madam Ainul. Okay,
Place two cylinder.
okay, nampak lebihan pigment tu akan ada pada tisu lah kan, dengan awak dah basuh dia tadi kan. So you don't want pigment from the broken cell to affect the results when you are immersing in these three different solution. Okay. Okay, so this is the result that you want. Okay, so the initial color of the solution before you immerse is all are clear in color. Okay, all are clear in color. So this is the initial color. Initial color, all clear. All right, and then you wait for
Let's stand for 15 minutes. 100% it. So
okay. So this is the result that you want. Okay, the result that you want to observe, the final color. So to record your result, the initial and the final color, you have to construct a table. Okay, you have to construct a table. So for example, when you are constructing the table, the first column you can uh, put all the different different solutions. So the distilled water with the 2 cylinders of turmeric, and then the 25% ethanol, and then lastly you have the 100% ethanol. Okay, so and then the next column is for you to record for each of these solution the initial color and also the final color. So the initial color, you know that all these three solution with turmeric in it, it appears clear. Okay, so for the final color, you want to record the result where for the distilled color, for the distilled water, the color remains clear. Okay, and as for the 25% ethanol, the color turns to light yellow, and then for the 100% ethanol, the color turns to bright yellow. Okay, light yellow for 25, 100% is bright yellow. So why do you think that there's differences in the color of the solution for these three different solution after you immerse the turmeric for 15 minutes in this different solution? Why do you think that there's different, the color turn into different, the solution turn into different color? Why do you think that? Kalau distilled water remains clear, kalau ethanol jadi light yellow, kalau 100% ethanol jadi bright yellow color. Why do you think influence? What do you think are the factors that influence the color to change to these respective colors? Kenapa? Tengok balik tajuk asal eksperimen ni. Tajuk asal apa? Investigating cell membrane permeability using turmeric. How do you relate, okay, the membrane permeability with immersing the turmeric inside these three different solution? Kenapa ada perbezaan color pada tiga solution ni? Cuba relatekan. Saya bagi clue yang macam saya cakap tadi, the turmeric are made up of cells. Okay, so cells, the cells are surrounded by plasma membrane. Okay, the pigment within the cell are also stored in a pigment. In an organelle. Okay, so the organelle also surrounded by membrane. So what are the component of this membrane? The plasma membrane and also the membrane of the organelle that stores the pigment. What is the what is the composition of the membrane?
Madam,
Yeah.
if I say the concentration of water in turmeric cell is higher than ethanol,
Kenapa tak the concentration of the concentration of salt inside the turmeric is higher compared to ethanol?
yes.
Okay. Pergi balik kepada asas. Saya tanya tadi, what is the composition of the membrane within cell? Plasma membrane and also the organelle that stores the pigment. What is the composition of membranes?
Lipid.
To be specific, lipid kita ada banyak kan? Kita ada triglyceride, kita ada phospholipid, kita ada steroid. Which one?
Phospholipid bilayer.
Phospholipid bilayer. Okay, so phospholipid bilayer forms the main structure of this membrane. Okay, so phospholipid is grouped under lipid. Okay, so what is the solvent that can dissolve lipid?
Organic solvent,
Organic solvent. So ethanol falls under organic solvent, is it not?
Yes.
Okay, so that's why when you immerse this turmeric into these three different solutions, distilled water, diluted ethanol, and concentrated ethanol, you will find that the solution will turn into different intensity of yellow color. Okay, so as you can observe, the 100% ethanol will turn to bright yellow color. Why? Because the solvent, in this case is the ethanol, will break or will damage the plasma membrane of the cell and also the membrane of the organelle that stores the pigment. Okay, so when the ethanol break and damage the membrane, the pigment will be released. Okay, so as you can observe, for the beaker containing 25% ethanol and 100% ethanol, the 100% will release more pigment because more membrane will be damaged. Okay, because the ethanol is much more concentrated. Compared to the 25%, the color will turn to yellow, light yellow color because the plasma membrane also will get damaged, but not as much as the one that are immersed in 100% ethanol. And so still the colors turn to yellow, but light yellow because the solvent is less concentrated. It is 25%. Okay, so less membrane will be damaged, tapi masih ada damage pada membrane tu, so that's why pigment from this damaged cell will be released. Okay, so so that one you have to understand. So pergerakan, the movement of the pigment, okay, from cell into the solvent is through the process of diffusion. So kita nak apply, we are going to apply our knowledge from chapter 3 lah kan, experiment 3, chapter 3, which is on plasma membrane kan. So you have to know the composition of the plasma membrane consists of phospholipid, which will dissolve and damage inside organic solvent, which is ethanol. Okay, so as for the turmeric that you immerse in distilled water, as you can see, there is no change in color because water is not the solvent for plasma membrane, so it will not give effect to the membrane integrity. Kan? So tak ada kesan bila awak letak turmeric dalam air, sebab air bukanlah solvent pada plasma membrane. So plasma membrane of the cell will remain intact and no pigment will be released, okay, into the solution. Alright, boleh faham
Boleh.
Okay, so so when you are discussing your result. Okay, so you must relate all this lah kan. Apa peranan ethanol kan, kenapa, apa efek dia kepada plasma membrane, kenapa ada perubahan colour, which is the pigment will move from the more concentrated region to the less concentrated region kan, from the cell, okay, from the cell to the solution. Okay,
Azman as
ada yang main kucing ke? Okay, alright. So can you so I hope that you understand. Okay, so discussion awak ni should not relate with the concept of pergerakan air kan, osmosis, hypertonic, hypotonic, isotonic solution. It does not related to that. Alright. So kalau iso, hypo, dia adalah melibatkan pergerakan air. Kan, pergerakan air keluar masuk sel in this different solution. Hypo, isotonic solution is not related to this experiment. Okay, because we are not using salt water or saline water or sugar, water that contains sugar. Okay, we are not dealing with solution that has different concentration of salt, okay, that will influence the movement of water. Kita nak tengok pergerakan pigment ni daripada dalam sel kepada luar sel disebabkan oleh plasma membrane to damage, disebabkan you all immerse dalam ethanol which function as organic solvent for the plasma membrane. Okay, so that one you have to include in your discussion. So you must refer to references, not just my explanation here. Alright. Okay, so you record your result in table form, and then you may include this diagram also in your result and discuss your result, okay, based on the what you have learned, okay, in chapter 3. Okay, so do your report for experiment 3. So the lab report for experiment 3 and experiment 2, you do it separately kan. So semua mesti ada title, introduction, the procedures kan, that you write in past tense, and then the result, and then discussion, and then conclusion, and then references, okay, to get the whole report. So please do it together. You have 4 to 5 members in a group, okay. So distribute the work together, and then when you are compiling the work, make sure you check dulu before you submit. Okay, so kerja orang lain akan mempengaruhi markah you all, and so buat sehabis mungkin sebab you akan contribute kepada markah orang, sebab ini adalah kerja berkumpulan. Jangan buat kerja tampil lewa lah kan, sebab you are responsible to other people's mark as well. Alright. Okay. So do you have any question before we end our session? I guess that's the end for the the explanation for these 2 experiments. Okay, so kalau tak ada soalan, I hope that you understand what you have to do for the report for both experiment. So I give you usually, okay, so usually the time given for you to complete your report is 1 week. Okay, so that's why S48 saya minta masuk juga kepada session ni, because when we have completed our experiment for these two experiment 2 and 3 ni, you have a longer time to complete your report, because then you have to submit your report after the holiday. So you have your exam week and then your holiday, and then you submit your report during week 8 after the holiday, okay, which is on Friday lah, Friday on week 8. So remember to submit your report on time in the Google Drive, okay, that I've shared with you. Okay, so saya nak share dulu sebelum kita end our session this lab schedule, okay. Okay, so ni adalah minggu ke-7. Okay, so this is week 7. Saya tak payahlah, saya tak stop dulu lah. Okay. So this is week 7. So week 7, week 7, you have completed experiment 2 and experiment 3. So this is the last video demonstration that we are going to observe, and for you to do your report. You have your test next week, and then you have um, your holiday, can midterm break, and then continue with week 8. Okay, so minggu ni sebab you all dah buat eksperimen ni minggu ni, so that one should be minggu tu free. Okay, tapi make sure to submit the report on Friday lah. Okay, and then with week after that, week 9, week 10. Okay, so this is the week that you have to secara dalam jadualnya you carry out the experiment and you write your report and you record your presentation. Minggu ni. Okay, minggu 9, 10 ni you do your experiment, you write your report and you record your presentation of the mini project. Okay, so for S47, you are going to present your mini project week 11. S48, you are going to present your mini project on week 12 on Friday. Okay, so the video, the video and also the report for the mini project, you have to submit 1 day earlier before the actual day of the presentation, at least 1 day earlier. So for example, group S47, your presentation will be on November, minggu start dengan 1 hari bulan. So you are going to present on the 5th of November. So by 4th of November, your video and also your report must be in the drive. Okay, must be in the drive by Thursday. Okay, kalau untuk S48, okay, you are going to present on week 12, okay, which is on the 12th of November, 12th of November. So you are going to submit at least 1 day earlier, okay, which is on the 11th in the drive. So during the presentation, I will play the video. Okay, so your video should not be longer than 15 minutes because after that you have 5 minutes of Q&A. So we have 5 group, we have 2 hours of lab session. So each group will have 20 minutes of presentation. 15 minutes is allocated for you to, for me to show your video presentation. 15 minutes for your video to be shown during the session, and another 5 minutes is for Q&A. Okay, Q&A. So Q&A adalah wajib sebab itu adalah salah satu pemarkahan yang akan diambil lah. Okay, so you can refer to the rubric in your buku workbook tu. Dah letak dah rubric untuk pemarkahan. You can refer as a guide for you to prepare your assignment. Okay. Same goes to the bio-D presentation. So bio-D presentation, video should not be longer than 15 minutes because another 5 minutes is for Q&A. Okay, so kedua-dua video ni dalam masa 10 hingga 15 minit sahaja. Okay, not longer than that. So S47, you are going to present on week 13. So week 13 is you are going to present on the 19th of November. So the video should be in the drive by 18. Okay. Same goes to S48. You are going to present on week 14, which is on the 26th. So your video should be in the drive by 25 on Thursday. So saya akan download and I will show the video during the presentation session. Okay, so walaupun minggu ke-9 ni kita balik pada mini project, walaupun mini project ni minggu ni diperuntukkan untuk you all do your experiment and do your report, doesn't mean that you have to wait minggu ke-9 baru nak buat. Okay, so you have the whole entire week of your holiday, the whole entire week 8, okay, and then week 9 and then week 10 before you have to submit your report. So please start now lah. Start after the exam. Saya tahu sekarang ni orang lupakan benda-benda lain. You are focusing to your exam. So after the exam, during the holiday, you can start doing your experiment and can start even to do your write up for the report. Okay, so tak payah nak tunggu minggu ke-9, 10 untuk buat. Okay, so you can start from your holiday to prepare for the mini project and also to prepare for the bio-D presentation. Jangan tunggu last ni baru nak buat. Sebab apa? Sebab minggu ni nanti akan dah ada banyak lagi assignment daripada subjek lain yang you all kena submit. Especially subjek yang tak ada exam pun akan buat exam pada minggu-minggu ni juga. Okay. So saya tak sure exam you are going to take your CTU kan? CTU dengan ELC kan? CTU dengan ELC tak ada final tapi dia akan bagi exam pada minggu-minggu ni. So make sure you don't wait until the last minute to do your assignment for the mini project and also bio-D presentation. Okay. Saya tak nak ada yang complain, oh madam, kami tak ada masa untuk buat report dan video ni secara elok je. Kan ada yang pas dia punya batch, dia prepare video ala kadar, tak explain pun the whole experiment, so dia akan affect your marks. Okay, so you have the whole entire weekend of your holiday ni, the whole entire holiday ni untuk prepare. Kalau ikutkan minggu ni free, we don't have any session. Okay, ini adalah session kita minggu ni untuk buat untuk kita buat eksperimen dan kita akan jumpa balik pada minggu presentation. So minggu 8, minggu 9, minggu 10, masa 2 jam hari Jumaat ni you should be free. Pukul 8, pukul 10 ni sepatutnya secara idealnya you sit down together in your group, okay, discuss and then do the experiment dalam masa 2 jam yang diperuntukkan, 8 hingga 10 tu. So kalau let's say awak peruntukkan sepenuhnya 8, 10 minggu ke-8, 8, 10 minggu 9, 8, 10 minggu ke-10, especially S48 ni ada access minggu ke-11 ni kan. So you have plenty of time to do. And then pada minggu holiday ni, so at least minggu ke-8 2 jam kan. 2, 4, 6 by a group of 5 or 4 people should be you should have plenty of time to do your assignment. Okay. So I hope that you do the mini project and also bio-D presentation properly. Okay. So do you guys have any question to ask before we end our session? Ada soalan? Tak ada So I hope that you know what to do for your bio assignment. Okay, so now you have your report for experiment 2, your report for experiment 3, mini project and also bio-D presentation. Okay, so just focus on your test first pada minggu ni untuk minggu depan, and then after that you boleh start dengan buat report dengan all of the other assignment. Okay, so if you don't have any question anymore, good luck for your test. Prepare sepenuhnya untuk awak punya test ni lah, sebab walaupun dia test, markah dia adalah 15%, which is banyak. So make sure you all boleh jawab kan. So kalau tak boleh jawab, dia akan menurunkan markah carry mark awak. Selalunya bila you get your final carry mark untuk 50%, yang akan tarik markah tu sebenarnya adalah daripada test. Okay, so make sure you prepare the test dengan sehabis baik lah. Okay, so thank you to all. So I guess we end our session. Thank you for your attention and participation. Okay, so you may leave the meet. Okay, Assalamualaikum.
Waalaikumsalam. Thank you, madam.
Waalaikumsalam.
Thank you, madam.
Right.